I am pleased to announce two very distinguished ophthalmologists and glaucoma specialists, Sahar Bedrood, and she is a practicing glaucoma and cataract surgeon in Los Angeles and also a big contributor to Glaucoma Today and INET, and also Dr. Tom Samuelson. And I think his awards speak for themselves as he's won the Junior, Senior, and Lifetime Achievement Award for the American Academy. So that pretty much speaks to his distinguished nature. So today we're going to learn a little bit about emerging glaucoma devices, and what a better audience for such a fun topic.
Thank you and welcome. We hope this will be a really rapid fire, exciting 80 minutes or so. We've got 8 exciting companies that are going to present their story, and then we've got a terrific panel that will query them on their story. So we've got 3 panelists. Ray, starting with you, why don't you just introduce yourself, our 3 panelists, and what your practice is like.
My name's Ray Brown. Am I on? And I'm in a practice in Atlanta. I'm a glaucoma specialist, and I'm the Chief Medical Officer for Sight Sciences.
I'm Davinder Grover. I'm a glaucoma specialist in Dallas, Texas.
My name is Yvonne O, and I'm local here. I'm on faculty at UCSF, also a glaucoma specialist.
Wonderful. So I think that we're going to go ahead and get started for the sake of time. Our first speaker is from Belkin Vision. Daria Lehman-Blumenthal will be presenting on Belkin Vision. Yeah. Wow. We're all serious here.
Very serious.
We got to get people in from that break time, too.
Okay, yeah. So maybe, we'll wait a few more minutes, or you want
Oh,
me
no,
to
we're going to start.
Yeah.
We have 8 companies to go through.
Okay. So let's start. I'm Daria Lehman-Blumenthal. I'm the CEO and co-founder of Belkin Vision. I'm here today to speak about direct SLT, DSLT, which is an automated non-contact therapy for glaucoma. It has a CE mark under the rigorous MDR framework, and we have started commercialization in Europe. It's a known fact that there is insufficient number of doctors to provide care to all glaucoma patients. One way to bridge this gap is with an accessible treatment that can be used by any ophthalmologist, as prompt lowering of IOP is key to saving vision. Today, eye drops are still the default initial treatment offered by most ophthalmologists. However, I think everyone here would agree that patient adherence is the biggest limitation to disease management. 3 years ago, the LIGHT study found that SLT is more cost-effective than drops as a first-line treatment for glaucoma. The European Glaucoma Society and the American Academy of Ophthalmology have both updated their preferred practice patterns to recognize or recommend laser as an initial intervention. The NICE UK are now guiding doctors to use laser before drops. With this consensus, we are seeing a shift towards a drop-free glaucoma management, which not only restores control back into the doctor's hands, but also reduces treatment burden for the patient. The question is whether we can actually implement this new standard of care with conventional SLT. And it seems that there hasn't been a practical solution to expand the use of SLT until now. Let's see how DSLT works. The laser beam is delivered to the trabecular meshwork through the limbus. The Eagle device is ergonomic with heads-up touchscreen display. We've removed the oculars. A gonio lens is not needed. Our proprietary algorithms map the limbus and track any movement of the eye. Once the start button is pressed, you'll see a 1 preview cycle, 1 treatment cycle, and treatment is complete. The procedure is so intuitive, any ophthalmologist can do it, resulting in many more patients being treated efficiently. DSLT has been evaluated clinically in a substantial number of patients, including a sizable randomized cohort, under the rigorously designed pivotal study called GLORIOUS. The study found that the DSLT has a meaningful IOP reduction and an excellent safety profile. As I mentioned, in November, we started control rollout in Europe with our first customer in Germany, Professor Burkhard Dick and his team. They have so far treated more than 120 eyes with no serious adverse events and satisfied patients. Apart from providing care to glaucoma patients in general, there is a growing recognition that lack of health care for the underserved populations is a public health equity issue that needs to be addressed. Eye care professionals should be able to offer their patients treatments that are not only effective, but are also affordable and accessible. We are confident that DSLT will meet this immense public health need as laser therapy is emerging as first-line choice for the growing number of glaucoma patients in the US and worldwide. Thank you, and I welcome you to visit our booth and experience the Eagle device firsthand. Thanks.
Thank you, Daria, for introducing us to this new technology. I will have Dr. Wu discuss with us, maybe start the conversation and ask some questions.
Sure, thank you. That was really exciting to see, and I definitely see the advantage of, rapid gonio lens-free treatment, and certainly the equity argument in terms of being able to deliver more care to more patients. What I'm curious about is, of course, with traditional SLT, we titrate the laser to the patient's trabecular meshwork. With this direct SLT approach, are you able to titrate the treatment, or do you start with the standard treatment?
Yeah, we start with the standard. We use 1.8 millijoule because of the laser beam is transferred through more tissues, so we increase the energy. There are no safety issues, either in the GLP study that we did or the GLORIOUS study. And maybe I can answer with a question and whether titration is correlated with treatment efficacy, and I think the answer is no.
Sure, I think that's part of it, but of course also the risk of IOP spike, especially in patients with various amount of pigment or the amount of laser uptake. Also, titration might be attractive in terms of, for example, there are now ongoing studies to look at low-dose repetitive treatment. So if there were some options for, I assume you could titrate it, right?
Yeah, you could use any energy that you'd like. Mm-hmm. But basically, we would use the default energy, and really there are the safety profile is excellent. So we didn't see any issue. We treated more than 100 patients already, and we had no issues with pigment or...
Yvonne, you make a really good point, though, because when most people think about titrating, they're speaking in terms of efficacy, and there is no really good data to say that we know what we're doing when we titrate SLT energy to get a better or worse outcome. But your point about safety is a good one in terms of using the right amount of energy.
Yeah, and I think also, right, in a clinical trial that's in a maybe perhaps more homogeneous population, as this is used across the world, there might be differences, right?
So for now in Germany, we treated, the doctors, they have treated more than 120 eyes, and we had no issue with pigmented eyes or light eyes.
Has there been any histology? I mean, I assume not, but...
Sorry?
Histology? Have you looked at the angle post-treatment?
Yeah, of course. Of course, in the clinical study, we had various exams we had to do after, but definitely they look at the angle. We see if there is any.
No. I'm talking about microscopic histology.
So we did a GLP study, a very thorough one with rabbits. We compared the DSLT with SLT. We used 3.5 millijoule. We used multiple times the same spot on the, this laser delivery on the same spot, and there are any safety issues, even with the highest energy that the laser can deliver.
And you've not had somebody who was run over by a car on the way out in a post-op visit. All right.
Daria, I will ask one question. One similarity I see is between this and direct SLT is similarity between CPC and ECP. And we see a difference in the amount of inflammation because you have to crank up the energy, and you have to, it's just a little bit different in the way it's
totally
applied.
different,
So I want to see if you have any concerns, or have you seen any differences as far as post-inflammation?
Yeah, so no, and we have to remember that the energy in SLT and DSLT is very, very low. In CPC, we are talking about 10,000 times the energy. Totally different procedures. They don't resemble each other at all. And the target is different, right? We are targeting
of course, the
trabecular
target is different.
meshwork.
But in the similar way that one is very direct and one goes through the sclera, it could be a difference in inflammation, I think. And we will see this, when it comes out.
Yeah.
I don't know.
Outstanding. We need to move on. So our next topic is Elios Vision, and it'll be presented by Dr. Johanna Gonzalez, who's the International Medical Director of Elios. Dr. Gonzalez.
Hello, good evening to everybody. My name is Johanna Gonzalez. I am a fellowship-trained glaucoma surgeon practicing in Mexico, and I also serve as the medical lead for Elios Vision. Okay, so I'd like to start by stating the relationship between glaucoma and cataract. As they are both related to aging, we are seeing more and more comorbidity. So there is no surprise that out of the 32 million people treated for cataract each year, up to 20% of them will have glaucoma or ocular hypertension, resulting in about 6 million people a year with both diseases. Cataract surgery offers a unique opportunity for the treatment of glaucoma. And even though the last 10 years have been transformative in this space with the advent of MIGS, there's still a lot of headroom for improving and optimally treating patients for glaucoma at the time of cataract surgery. Ideally, a treatment for glaucoma during phacoemulsification would be safe, able to provide effective IOP lowering, and also desirable characteristics would be for it to be highly adoptable, so that it has a short learning curve, and because most patients won't have the chance to be referred to a glaucoma specialist for their cataract surgery, and have a long-lasting effect. Additionally, another desirable characteristic is that it wouldn't need a retained foreign body to be left behind. Elios is a procedure that addresses the increased resistance to outflow characteristic of patients with glaucoma. It uses excimer laser to create 10 microchannels in the trabecular meshwork up to the inner wall of Schlemm's canal to increase the natural outflow. Excimer laser has the advantage of being incredibly precise, and it has also the advantage of creating minimum, it's a non-thermal laser, so it has minimum inflammation and minimum trauma, not disrupting the natural physiology and anatomy of the angle structures. When talking about the equipment itself, it consists of a small console, a foot pedal for the surgeon to operate and deliver the laser, and a single-use handpiece that will enter the eye. Let's see, can we play a video here? Yes. So here we can see the surgery in a real eye. The surgeon will enter the eye through standard phaco incisions. He will approach the trabecular meshwork, and with very slight pressure come in contact with it, then deliver the laser with the foot pedal, and as you can see, bubbles are formed in the site of each microchannel creation, which reassures the surgeon of adequate microchannels. At the conclusion of the surgery, phaco is completed as per routine clinical practice, and no additional follow-up or post-op treatment measures are required. Noteworthy is that this technology has had a CE mark for the past 8 years, since 2014 in Europe, and thousands of patients have been treated with it. There's about 10 clinical studies published in peer-reviewed journals that add up to about 600 patients treated, reporting an IOP reduction ranging from 29 to 43%. But more striking are the changes in medication use, with publications reporting up to a little bit over 80% of patients being medication-free at 1 year. Recently, coming out of Zurich, an 8-year-old follow-up study was published. They treated 161 eyes, and they reported an IOP decrease greater than 20%. But what's striking is they saw a significant change in medication for up to 4 years and remained below baseline for the whole 8-year period. Notably, they also reported a secondary surgical rate for invasive glaucoma procedures at very low, at 3.7%. Another smaller study, retrospective, coming out of Spain, that treated 34 eyes with primary open-angle glaucoma with a combined procedure, reported a decrease in medication down to 0 in 82% of their patients, being medication-free at 1 year, with an IOP reduction of 22%. So as a summary, cataract surgery offers a unique opportunity to access angle structures that are amenable to the treatment of glaucoma. An ideal treatment for glaucoma at the time of cataract surgery would be safe and effective and comply with the criteria we know that defines mixed procedures. With the publications that we have available right now, Elios has shown an effective IOP lowering effect with a favorable safety profile without the need of having to leave a foreign material behind. It is currently undergoing an IDE pivotal trial in the US as a combined procedure. Thank you.
Thank you, Dr. Gonzalez. Dr. Grover is going to start the discussion.
Sure. Dr. Gonzalez, very nice presentation. You know, they say the best and the happiest glaucoma doctor is one that moves every 5 years, because the curse of long-term follow-up is just, it gets you. And that's true with traditional glaucoma surgery, that's true with MIGS and this minimally invasive surgery. You know, what's unique about your presentation is a lot of these talks that we've heard, when they're novel surgeries, we don't have the benefit of more than a year follow-up or 2 years follow-up. And that paper you presented out of Europe showing up to 8 years follow-up, I mean, can you talk to us more about that follow-up and how many patients you had, and how are they doing as far as med-wise and the insight that you have with that 8 years of follow-up?
Yep. As a matter of fact, I just presented one study, but there are actually 2 studies with 8-year follow-up from 2 different study groups. One of them with 161 eyes and another one with 164 eyes. They both follow the patients looking at efficacy and safety for the whole 8-year period, and they both found that the IOP lowering effect was still patent, or English is my second language, so I'm trying to find the word, but was still present up to 8 years. The study coming out of Zurich, as a matter of fact, looked at medication use, and that was one of the most, I think, important factors that they looked at, because it did remain below baseline for the whole 8 years. So even though it was just significant for the first 4 years, it remained below baseline. So I think anybody who actually treats glaucoma patients know how much of a challenge it is for them to be using multiple drops or even 1 drop. And we know from studies that even going from 1 drop to 2 drops can drop the compliance rate up to 60%. So I think as glaucoma surgeons and as doctors looking at glaucoma patients, we're focused on pressure, because we know that's what's gonna maintain patients' vision. But when we kind of put ourselves in their shoes and look at what quality of life is, medication use is a big part in these patients.
I can barely give a presentation in my native language, let alone a second language, so you're doing an amazing job. But what were the patient profile? Were they, I mean, these patients that you have the 8-year follow-up, were they more the mild patients that have been on drops for just a little while, or can you give us an idea of who these patients were?
So looking at this study, it is a retrospective study. They did have multiple diagnoses, not only primary open-angle glaucoma, but there were secondary open-angle glaucoma, such as pseudoexfoliation and pigmentary glaucoma in the group. And there was most patients were mild to moderate, but there were actually patients with severe glaucoma. As we know, dropping pressure 20%, 30% as per the guidelines is also important even in patients that have advanced disease, but most patients in the studies are mild to moderate. And something I forgot to mention but I think might be important, the study that's coming out of Spain actually has now extended its sample to 116 patients, and they will be presenting their results later this year at WGC.
Ray, I think you had a question and maybe a comment.
Yeah, what about pseudophakic patients? This would seem to be a great treatment for pseudophakic patients.
As a matter of fact, with the experience that we have coming out of Europe, there's actually a publication that looked at standalone versus combined cataract surgery, so that there could be a differentiation of the effect that phacoemulsification alone can provide. And both groups had a statistically significant decrease in IOP. In the group that had a combined procedure, there was a numerical tendency to have a higher effect or a more potent effect. So there is publications with standalone experience as well.
Sounds good.
Thank you so much, Dr. Gonzalez, for that presentation. We are going to move on. It is my distinct honor to present Malvina Eidelman, who is the Director of the Office of Health Technology for the FDA, and she has presented all day, and we look forward to her presenting emerging glaucoma devices in 5 minutes.
Thank you. So I was given this formidable task to summarize everything about emerging glaucoma devices in 5 minutes. So I welcome all of you to join me in this helicopter overview. So what do we have today? Everything in glaucoma diagnostics that is legally on the market in the US is on the slide. We have tonometers, we have fundus cameras, we have devices for functional tests, SAP, SWAP, FDT, devices for structural tests, GDX, HRT, OCT. Yes, I'm from New York and I speak fast. And what do we have from Glaucoma Therapeutics? We have surgical instruments, we have lasers, YAG, argon, neodymium, and we have only 2 kinds of implants, aqueous shunts and MIGS. Now, with all of this, what are legal indications for use? The only things that is legally approved for use in the United States today are devices for decreasing in IOP, devices for qualitative imaging, quantitative measurements, but do not mention specific disease, and those that are approved for aid in the diagnosis of a specific disease. We do not currently have anything approved for screening, diagnosing, or treatment of glaucoma. So, how do we get there? How do we close the gap? I believe there's 3 things that the whole medical device ecosystem needs to concentrate on and to work collaboratively to harmonize terminology, address health inequity, and maximize AI/ML. In our recent publication from CCOI Glaucoma Group, we wrote, "The lack of a unified gold standard for glaucoma diagnosis is perhaps one of the biggest challenges that the field faces." And as we were writing this, we were quite certain that we were not unique. There were many, many publications all around the world that cite lack of terminology consensus as the stopping block to our progress. Everybody in this room is well aware of the inequities in glaucoma care. The disparities in glaucoma impact prevalence, medication adherence, follow-up, and surgery. We at FDA, at CDRH specifically, have identified advancing health equity as one of our strategic priorities. To that end, we have added 2 recent guidance documents to the previous multitude of guidance that are now focused on advancing health equity. One of them specifically talks about improving enrollment of underrepresented participants, and the second one is about fostering innovation that addresses disparities. Now, while these are drafts, I welcome all of you to get online and let your thoughts be heard. We will incorporate everybody's comments as we proceed to finalizing these documents. And throughout the day, you keep hearing this important message. Digital health technology is everywhere. You can't escape from it. If you want to move anything in the device space, you need to incorporate digital health technology. And we're incredibly fortunate that Joel Schulman, under the auspices of Collaborative Community of Ophthalmic Imaging, has taken on the task to come up with consensus so as to advance AI, ML, diagnostics, and therapeutics in the field of glaucoma. So, where are we going? We will adapt standards. We need to come up consistent terminology. We need to reduce bias, address equity, and maximize utilization of AI, ML. And I think I was under 5 minutes.
We are going to have you start this panel discussion.
So, Malvina, we go back a long way. And you've been an amazing director, and my experience has always been in trying to get devices approved. And all of your points are so on target. From my standpoint, the thing that I have the hardest time with is how long it takes. Is there anything that can be done to speed up the process?
Well, it actually takes very short time if you have a very good submission.
Yes. You know, I knew that it was my problem, but you know,
we actually have statutory deadlines for every type of a submission, and now we just went under MDAFMA 5 authorization, and the timelines got even shorter. So if you look at the performance, we adhere to every single timeline.
Oh, absolutely, and are there things that you feel that you approved too early? Has that ever happened?
That you what?
That you approved before their time, or you, looking back, regret approving?
So, you know, that's a very good question. We have been moving towards utilization of RWE and maximizing post-approval studies, and actually trying to see where sometimes we can move the needle so that while we have minimal evidence for safety and efficacy at the time of approval, we can put some of the regulatory burden on the post-approval. So we have done that in a number of places, and I'm sure many in the audience are well familiar with a particular mixed device that shall remain nameless for the moment, that you know, the issue was identified during the post-approval study, and that's what those post-approval studies are for. And we have then been able to utilize the data collected during the post-approval studies to modify the future of the whole MIGS industry.
You know, that's a great idea. And I applaud those efforts and hope they continue.
Any other panel members want to ask a question? Go ahead, Yvonne.
Sure. I was definitely very interested in your discussion about sort of bridging the digital divide. And I'm curious what advice you would give to companies about trying to ensure that their technologies are addressing them beyond just, you know, incorporating a more diverse population in the clinical trials. But, you know, when I think forward about some of the devices that we're going to be talking about, you can imagine a situation where perhaps those in under-resourced settings may not have access to those devices?
Yeah, so that's a great question, because it gets exactly at the two guidance that we just that I was just presented on the slide. So glaucoma especially is a poster child for that. We are doing currently a deep dive where we're going back and looking at every approval per category, so per device type. So we're going back and looking at all of the clinical data that was submitted in support of glaucoma devices on the market today, and trying to stratify how much of that really was in the population and where the glaucoma patients are in the US, and we're trying to compare. And that's going to be our foundation, and then we will encourage all of industry to get beyond that, to make sure that at least 95% of future submissions, and future, I mean 2-3 years, actually study the population for which the device is intended. So we hope that by 2025, clinical trial for a glaucoma device will be very representative of the population of glaucoma patients in the United States.
Fantastic. Thank you.
Thank you, Dr. Edelman. Our next speaker is Dr. Alan Crunkleton. He's the President and Chief Executive Officer of IOPtic.
Well, thank you. Thank you for having me. I'm delighted to be here to tell you about IOPtic and tell you about the device we're developing for glaucoma patients to measure their intraocular pressure. The device consists of a very small pressure sensor that's permanently implanted and a handheld reader. The pressure sensor is a mechanical diaphragm, which is a very thin membrane that changes shape as the pressure changes. The handheld reader has a light source that illuminates the diaphragm and a camera that takes a picture of an interference pattern that reflects back from the diaphragm, which gives us the pressure very accurately to within 1 millimeter of mercury. You see the sensor is attached to the iris, and there are no implanted electronics. That's what sets our device apart from other implanted pressure sensors. And here's a complete product, which consists of the pressure sensor implant, the insertion device, and a handheld reader. The pressure sensor is implanted during a combination procedure, either cataract or glaucoma surgery. So for example, once the cataract surgery is complete, the implant is inserted through the same incision that was used for cataract surgery, and attaching the implant takes less than a minute. The patient then uses a handheld reader to collect data, which is shared with the doctor's office using a cloud-based connection, so that once a month, the clinician can review the data. And this is the data we've never had. With this data, we'll be able to monitor whether the treatment is working and whether the patient is complying. And here's more of a deep dive on the sensor itself. On the left, the sensor diameter is 500 microns, and it's mounted on a clear PMMA housing that gets attached to the iris, and the largest dimension of the implant is less than 2 millimeters. These images here illustrate how the interference pattern changes as the pressure changes. So we record images of the interference patterns, correlate those with the pressures, and that's our calibration curve. As far as bench testing, the center plot shows how the sensor achieves a sub 1 millimeter accuracy, 1 millimeter mercury accuracy, even when subjected to a temperature range that exceeds what we experience in our daily life. We also did extended aging, accelerated aging, which that showed that the baseline drift was less than 1 millimeter of mercury per year for a 5-year equivalent. The right plot shows data for after 6 months in rabbit eyes, and that showed we measured a precision of less than 2 millimeters of mercury. Other testing we did, ocular grading, we measured central corneal thicknesses, and all in, it just told us that the implant would be very well tolerated. We believe the reason why there's not already a product like this on the market is because no company has had the technologies to deliver on all of these requirements. I've talked about how we deliver high accuracy and ease of implantation. For the patient, there's no contact lenses. There's no disposable probes or goggles that these often elderly patients would have to deal with. And as far as the handheld reader, it's easy to use. It takes the patient literally just a few seconds to take a reading. As far as durability, with no implanted electronics, the implant will last for the life of the patient. And as far as affordability, using MEMS manufacturing techniques to make thousands of these sensors at a time, one wafer, and using off-the-shelf components for the rest of the system, the cost is low, giving us a price that'll be affordable to the payers with margins that'll be attractive to investors. Finally, I mentioned that all the work so far has been funded under NIH, SBIR, phases 1 and 2. Now we're fundraising to complete the development and do first-in-human cases. And with that, thank you.
Thank you. And Dr. U's going to start the discussion. And a reminder to the audience, if you have questions, feel free to text them in, please.
Thanks for that presentation. I am curious about, in terms of IOP monitoring, with the advantages of home monitoring and also allowing the clinician to get more information that they otherwise are definitely going to miss is a huge advantage. And I think this kind of technology will bring us to that era. In terms of, maybe you could talk a little bit about a few questions that I had, which were about how the device is actually implanted on the iris. I think I heard you mention that's a PMMA material, or how the sensor, I should say. And then also how you think IOPtic is going to line up compared to other methods of home tonometry, such as eye care, home eye care.
Yes. And your first question was?
Just about how the device is actually implanted on the iris.
Yeah. So what we did was we looked at other devices and said, what has a really good track record as far as adverse events, what have you. And what we did is we basically copied a very well-proven way of attaching to the iris. And what we did was just make it very easy to do. And that's about all I can say about it, but that's, okay. It's something that, and another important point is that if you take the envelope of an anterior IOL and our device fits completely within that envelope, so should suggest that adverse events should be minimal.
Okay. And then my other question was just about, this is...
Where do we fit?
Yeah, where do we fit? Because, I'm trying to imagine myself, certainly a home eye care tonometer is not necessarily the most user-friendly for an older patient. I can imagine a situation where getting a reliable reading using that device might be more difficult, especially depending on how the... I didn't quite completely understand, how orthogonal, for example, does the reader need to be to the sensor, for example, to get an accurate reading. I don't know the details of that. But you could imagine that this technology might be easier for someone to measure their IOP, but
right.
Yeah.
Yeah, so as far as ease of use, I think it helps not having to have anything extra like contact lenses or goggles and that kind of thing. But as far as use of the device, you literally just put it up to your eye and look at a little red dot. You press the button and that's it. So it takes just a few seconds. In fact, I point out that we did this with rabbits that were not sedated. So it's just a matter of sort of waiting until they get to the right place and press the button and you take a picture.
Dr. Kunkelton, a question from the audience pertaining to...
I'll also mention one other thing, that because you asked about how do you compare with eye care home and those types of devices. It's important that we're measuring the IOP directly. We're being in the intraocular fluids and measuring the pressure directly. I always like to say if you're trying to measure over the cornea, it's like trying to measure the pressure in your car tire by tapping on the tire or measuring surface changes. It's like, yeah, it can be done, but it's difficult and it changes as the weather changes, as it ages. It's not a direct measurement. You're limited by the accuracy.
The question from the audience pertained to how you use the information you acquire. So, for example, a patient with diabetes knows their blood sugar. A patient with hypertension knows their blood pressure. Do we share this information to the patients? Are they the first recipient of the data, and should they be? Maybe you comment, and then Davinder, if you want to give your opinion, and then we'll move on.
I think the truthful answer at this stage is that we don't know. We've never had this information. So, there's all the questions of how often will the data need to be taken, how will it be used. There's a lot of questions there.
Davinder, do you want the patient to have?
No, I agree, but I think this is there's certain things that will give us just tremendous insight into the disease of glaucoma. And anybody that's taken care of glaucoma for a while knows that what we consider the gold standard, gold 1 applanation, is not the best. And I can say that at a glaucoma meeting and not get tomatoes thrown at me. But that's not always the case in some meetings. That's the holy grail. People are very tied to gold 1 applanation. And it may not be the best way to know the pressure. And we all have patients that we think are controlled on gold 1 that are still getting worse. This, I think, any kind of method to get direct pressure measurement of the eye is going to give us tremendous insight into the nature of this disease that's still plaguing us all and that we can't have a unified definition for. We don't understand what's happening sometimes, and this, I think, will be a tremendous addition to our knowledge.
What you're really getting at is, what is the value added? And that we have a lot of work to do on that. We all know that overtreating is going to lead to higher costs, but we haven't quantified that exactly. We haven't quantified, how much, what is the benefit to the patient? What is how much will it affect the patient's compliance, for example? And all those things, we know that there are going to be good results, but we don't know what they are yet. That's the reason we're already planning for our clinical trial to collect that type of data directed at reimbursement.
Thank you so much.
I didn't answer Tom's question, actually, but about whether the patient should actually know that data. I think they should. I think the more we engage patients, the more they know about their health, their blood sugar, their blood pressure, and their eye pressure, the more they're going to be engaged in this battle.
Yeah, I agree. I absolutely agree with that. We're going to move on to the next speaker, who is Sean Iantrek from Iantrek, and he is the CEO and founder.
Thank you. Thank you very much for inviting me and for this great panel. It's actually great to see so much innovation happening in the field of glaucoma. I also want to thank Adrian and Andrew for inviting me back. And I would like to talk about 2 technologies that we are developing in the mixed space. To continue the story, last year, Bob Weinreb was on that same forum, and he presented on one of the technologies. And I would like to give you an update where we stand, and then to introduce something else we're doing, and hopefully get some good feedback from the panel. The first technology is really re-looking at biotissue. I think in ophthalmology, we're not new to biotissue. In fact, in glaucoma, we've been using biotissue a lot when we cover tubes, sclerografts, sometimes we do cornea. And one of the benefits of biotissue and donor tissue is that it's quite bioconforming. It is not hardware. I know we're looking at all these devices, but today you have an IOL, a CTR, a couple of mixed stents, and at that point, I think there's a lot of hardware that goes into those patients. So one of the benefits of the tissue is that it's really non-hardware, and it can give us some of the structural benefits that we're aiming with the hardware without putting a foreign body. I think Movina mentioned about what can happen with foreign body, and I think one of the benefits of using tissue is that some of those could be avoided. One of the benefits of sclera, when we look a little deeper, is that it is one of the more porous and permeable tissues, especially hydrophilic-wise. So it can let a lot of things through. And also, when we look at the science of implants, one thing that comes up is we talk about fibrosis all the time. And it turns out one of the first, second, and third probably all drivers of fibrosis is the mismatch of the structure of the stiffness of the implant to the surrounding tissue. And that's not only true for glaucoma and ophthalmology, this is across the board. And one of the benefits of using biotissue, especially in a homologous way within the vicinity of the surrounding tissue being the same, is that there is almost no mismatch. Now, whether that really pans out, we're supposed to see, but definitely sticking something hard in a soft surrounding tissue would be associated with a lot more fibrosis. And we all know that, not from ophthalmology, from almost every other implantology and science of implants. The good thing about also sclera is when we look at some of the studies, it's quite permeable. In fact, it's more permeable than cornea, which definitely allows for application in aqueous drainage clinical procedures. So, what can we do? Can we improve our microinterventional instrumentation to be able to leverage the new attributes or the more kind of rediscovered attributes of tissue? And again, they can be applied in different areas. For example, supraciliary space, we do want to tap the potential of the suprachoroidal outflow, so that could be one place where tissue can give us the structural support to open up the cyclodialysis and to increase drainage. It could be in areas of bleb rescue. We know there are sometimes patients with hypotony, and maybe that can be used ab interno to do a more occlusive application of the tissue so that you can prevent hypotony, or sometimes to lift a flap that's very structurally adhered and the bleb is failing. Also in retina, there are now quite a few cases of optic disc with serous retinal detachment that are handled by using tissue and scleral tissue to occlude them. And this is a great application because in all of these, the tissue can give us the benefit we need without any foreign body hardware, as long as we have the microinterventional instruments to allow us to handle the tissue, because these are different than what we currently have. So just in time for this meeting, the BJO was very kind to publish our one-year data of how we can use tissue in a microinterventional way for stenting or for opening up the supraciliary space. And we showed 12 months data on the first patients that were very encouraging. And they also showed very good biocompatibility of the scleral tissue. But today, I promised I'm going to talk about something different, something that we haven't disclosed and never presented before. And this is our second technology for next-gen canalotomy, or T-Rex, which we call. That name may change, but once we hire marketing people. But we call it T-Rex because we're basically addressing both the inner and outer wall, especially on the inner side without cutting, by using trabeculorhexis. We know the Schlemm canal is actually 360. It's not just one wall. And right now, with goniotomy, we're pretty much addressing the inner wall of the Schlemm's canal. Now, what about the outer wall? Actually, 3D microcomputed tomography shows that in glaucoma, 24% of the collected channels are occluded from the outer wall. And we have debris and involutions that actually prevent those channels from draining. Now, how can we address that? Is there a way for us to really tackle that and open up those channels of the outer wall so that we can improve outflow? So we developed a microinterventional device using nitinol superelastic memory shape technology, where we can address both walls, the inner and the outer wall. The inner wall is the trabeculorhexis by blunt dissection of the trabecular wall. And then, because we have a very firm instrument, it's not a catheter, it allows us to exert pressure and to modify the outer canal wall to also clean up and really open up the collected channels. Carol Torres actually did a study where she looked at the single wall versus the dual wall intervention, and there was much higher outflow facility increase with the dual wall intervention versus a single wall, and this is in cadaver eyes. So we're very excited. We've actually developed that instrument to be also handled robotically. Actually, you can see here it's attached on the robotic arm that we have at New York Eye and Ear, which allows us to use it micro-interventionally with very high precision. The surgeon usually has about 100 micron precision. The robot is at 1 micron. And we know the trabecular meshwork is small, and this is where we need to do a better job at really handling that instrumentation in the eye in order to improve our precision and know that we implant stents and hardware where it needs to go. Thank you.
You are presenting some amazing work. Davinder, I'll ask you to comment or maybe start the discussion as well.
Yeah, Sean, thank you for that great presentation of a lot of exciting stuff. I never fail to learn a tremendous amount whenever time I hear you talk. So thank you for sharing that and your innovation. It took us about 50 to 60, maybe even 70 years to understand wound healing in the subconjunctival space. We still don't understand wound healing in the angle, after angle surgery. And I think we know even less about wound healing in the suprachoroidal space. There's probably not many people in the world that have the experience of living in that space that you do. Given your experience with all the shunts and being in that space, what insights do you have on just in general wound healing in the suprachoroidal space, and then also how this biotissue is going to possibly be more favorable in that environment than other materials that have been invented or are being investigated right now?
Well, this is a very difficult question, and I'm sure we can talk about this for a long time. I think wound healing is part of every tissue, even in the canal. I'm sure we're going to find some wound healing differences. I don't think we know and understand the suprachoroidal space. I can also argue that maybe it's not even a simple question of outflow. When you look at some of the studies from the early 80s that were done, and they looked at outflow, it actually turns out a big part of the outflow enhancement that we think is outflow enhancement actually comes from decreased production by detaching the ciliary body. So again, I think we're very challenged. We need more imaging. I think the work that we're talking about, imaging, is very important. I remember when we were developing Lucentis, and when we first started the trials, we didn't have OCT. So I think the advent of OCT in retina has really improved a lot. I think we're in the pre-OCT age in glaucoma. We talk about enhancing outflow, but when you talk about that with a patient, I don't know what their outflow is before going to surgery, and I don't know what their outflow is after coming from surgery, and whether they need one stent or a suprachoroidal enhancement or a trabecular. So unfortunately, I think we are where we are. We have to get more imaging, and we have to understand, maybe figure out how to measure outflow to understand how that works. And also, I remember with the suprachoroidal work we did, it's very difficult to see fibrosis. We know there is encapsulation. By all means, there is always encapsulation. But one thing I can tell you for sure, when I look at all of the literature in implants, if we match the tissue to the surrounding tissue, the implant to the surrounding tissue, you're going to have much better fibrotic profile. And that's pretty much across 20, 30 studies that were done all around. So if we want to do that, that's a good strategy. And I think that's why we have now more flexible implants like the Amet with a flexible plate that I think is in a good direction. But I wouldn't claim that we understand it. We're kind of poking fingers at the space. Unfortunately, that's kind of, you start that way, but when you have a therapeutic or a new device in that space, it leads to innovation and drags along the imaging and understanding. And I think hopefully we'll be there as we put new therapeutic interventions and new devices. We'll learn a lot more.
Thanks, Sean. Unfortunately, we're out of time for this session, but outstanding stuff. Our next speaker is Dr. Lawrence Marsteller from Radiance Therapeutics, and while he's walking up, Ray did have a comment that you can make.
So Sean, you emphasized the T-Rex improves flow through the collector channels. How do you know and how do you do that?
So I wish I can see it. I wish we can even image it in a very detailed way. The way the studies were done by Dr. Torres is really measuring outflow in a very standard way, just like it was measured with some of the MIGS implants the same way. So we had one group of eyes which underwent single canal, inner canal wall procedure, and the others dual canal wall. And basically there was a statistical increase in outflow, but it's very hard to measure where the outflow is going and how that works. And I think this is only a dozen eyes, so I think we definitely need more data. Yeah.
Thanks, Sean. Dr. Marsteller.
Thank you. This is Radiance Therapeutics. Our mission is improving glaucoma drainage surgical outcomes. Our innovation addresses the unmet need in moderate to severe patients. Those are the patients who are actively losing vision and require drainage surgery. That could be the trabeculectomy or the new transscleral MIGS devices. This surgery has an unacceptably high failure rate from post-surgical scarring. We can see on the left a functioning bleb with the aqueous humor flowing, and in post-surgical era, that bleb can become scarred, restricting the flow with elevated IOP. Approximately 50% of the patients undergoing trabeculectomy can suffer with problems with scarring of the bleb. This study from the BMJ showing that at 3 to 4 years, there's an almost 50% scarring rate. In the transscleral MIGS, the success rate at 3 years can be as little as 47%, depending upon the study. We believe the solution, and we're going to show some clinical data here, is the Radiance ophthalmic applicator. The mechanism of action is beta irradiation. Beta therapy is well documented antimetabolite effects, downregulates tenin capsule fibroblasts via cell cycle controller P53. Clinical trial outcomes data shows significantly reduced postoperative scarring, maintaining low IOP. There's really a wealth of data on this therapy from a number of sources. This innovation comes from Moorfields Eye Hospital and University College London, and some of the studies I'm just going to show you were funded by the Wellcome Trust. In all, there's been 4 randomized controlled trials already completed and published. In Kerwin et al., this is a large study enrolling 320 patients undergoing trabeculectomy. In the placebo arm, which we wouldn't do anymore since the innovation of mitomycin C, approximately 50% scarring of the bleb by 3 to 4 years. But in the beta intervention arm, we see that the persistence of the bleb without scarring is 90% between 3 and 4 years. This is a notable increase in efficacy. In a follow-on study, this is Cook et al., enrolled 117 patients undergoing trabeculectomy. They were randomized head-to-head, either the current standard of care for scar prophylaxis, mitomycin C, or they received beta therapy. The odds ratio reported in this study at 1 year were that beta therapy eyes were 3.2 times more likely to have a functioning bleb versus the mitomycin C treated eyes, and 5.6 times more likely to have an interocular pressure less than 16 millimeters of mercury on no additional medical therapy versus the mitomycin C treated eyes. On average at 1 year, the beta therapy eyes have an IOP 4 millimeters lower than the mitomycin C treated eyes in this study. Most recently, Moorfields Eye Hospital has published a 20-year follow-up from a chart review of patients undergoing trabeculectomy with beta therapy. They have unprecedented data reporting 92% persistence of the blebs at 10 years and 83% persistence of the bleb at 20 years. This is going to allow for the 10-10-10 goal, which is a 10-minute surgical time with an IOP outcome of 10 millimeters, and we often see this initially with the placement of transscleral MIGS. This is now the final piece of that puzzle, which is lasting for 10 years or more. This therapy is now available electively at Moorfields Eye Hospital, and it is our hope that we'll be able to take this to patients around the world. The value proposition for the patient is that beta therapy is a clinically demonstrated enabling anti-scarring technology for all drainage surgeries, allowing for maintenance of low IOP. I'd just like to call out Keldev Singh, who I believe is here, who's been with us through thick and thin, and our thanks to him, to Keith Barton, and to Ike Ahmed, our advisors. Speaking of Ike, I would refer you to the recent beta review article in Glaucoma Today magazine. Thank you.
Thank you, and Dr. Brown's going to initiate the discussion.
So that was very interesting, and obviously maintaining blebs is a key thing for the refractory patients. This is applied at the time of surgery. It's done, you know, you do your trabeculectomy and then you put this on?
Yeah, so this is done, these were done at the time of surgery, just after closing the conj.
Can it be done later?
So there's actually a good study out of the University of Arizona for pterygium treatment, which has been using beta therapy for 100 years. They found that the optimal time was within 6 hours following surgery, so at the time of surgery and afterwards. That probably has to do with activation of fibroblasts. The mechanism of action is a double-stranded DNA break, and if you activate the fibroblast, it uncurls its DNA and allows for more efficacious application of the beta. I think now, and this is just an opinion without data, is that so many patients have inflammation from eye drops that you may actually be able to do this actually in advance of surgery as well.
How about complications, hypotony, bleb leaks, wound dehiscence, stuff like that?
So there's 100 years of beta therapy history for pterygium, and within that rich literature, you can find case reports of all of that. But as a percentage of the patients treated, it's all of these, in these particular studies, showed no side effects. And overall, it's known to be quite, historically, it's been shown to be quite safe.
Dr. Marcel, I don't think there's a person in the room who wouldn't think we could improve our antifibrotic treatments. Do you know if there's any transscleral cilioablative effect from beta radiation? Is there any effect on the ciliary body beyond its antifibrotic effect on the subtenons and conjunctival
That's a very good question. So there's 3 types of radiation we all remember from our first physics, X-rays, which are very penetrating, beta, which only actually goes a couple of millimeters, and then alpha, which goes even less. So beta really does it at the doses that are being given here, and these are sublethal doses. This is not like oncology where you're giving much higher doses. At these low doses, there's less than 4% dose to the lens, and even less for anything behind that.
That's a question.
Davinder?
Do you see this as a replacement for mitomycin C or a supplement to mitomycin C?
So in the in that second study I showed, that was a replacement for mitomycin C. There's another study that I didn't show, El Mazar et al. Out of Egypt, where they were adding it to mitomycin C and getting, with the addition of mitomycin C and beta, still showed better blebs and better pressures. So this could be, you know, I would imagine that conservative surgeons would probably start by adding it to their mitomycin C, and if you were getting good results, you might then be able to use beta only.
Are there differences in appearance of the bleb 10 years out, 20 years out with beta radiation?
I can't speak to the bleb appearance 10 and 20 years out because I wasn't part of that study, but there is this, in publication, El Mazar paper, which shows that blebs are much nicer looking, and specifically without vascularization or thinning.
Okay, thank you so much. We're going to move on to Sanoculis. If you can walk up here. This is Nir Israeli, who is the Chief Executive Officer of Sanoculis.
Well, hello to all of us. Thank you for the opportunity to present Sanoculis stentless novel technology. I hope during the next few minutes to show you how innovative this technology is. I will start that last September we have launched the MIMS technology with Bosch and Lomb at the ESCRS, and the market traction that we gained was a very nice one. Let's have a look on the technology itself. Our system is comprised of a foot switch, a controller, and disposable handpiece unit, the white part that you can see, with a tiny trephine that goes out and backward automatically while rotating. The procedure goes like this. The physician prepared the eye for the surgery, inject MMC and a little bit visco material, then fill the AC with visco, and then introduce the device to the angle of the eye. No need for gonio lens. There is a mark that helps you to guide and locate the device. Press the foot switch and create the sclerostomy to drain the aqueous to the subconscious space. The piece of tissue remains inside the trephine, which makes the device a disposable one. We have operated on 120 patients, 20 were combined with cataract, and the highlights of the 1 year and the 3 years results are impressive. You can see that the IOP was reduced by 38% at 1 year and by 39% at 3 years, and the average number of medication by 85% at 1 year and 69% at 3 years, with no major intra and postoperative complications. This is another view of the results. The above one is the IOP graph. You can see that the IOP was reduced from high levels of 27.9 to 17.3 at 1 year and 17 at 3 years. And again, the average number of medication was reduced significantly at 1 year and 3 years. In terms of success rate, where these results place us. So you can see for yourselves, MIMS is definitely in a good place when you look on the playing field and when you see the current numbers and results. You can extrapolate and understand where will it lead us, and we are at Sanoculis are very optimistic. This picture proves the concept of MIMS. You can see good example of channel at 1 year and then at 3 year, the same patient with great results compared to the baseline. Patient arrived with 32 on 1 med, and after 1 year, 14, 0 meds, and 3 years, 14.5 and 0 meds. Another example, you can see another patient, very nice result, arrived with 22 on 2 meds. After 1 year, 12, 0 meds. You can see clearly the channel. And after 3 years, the patient was on 9.5 and 0 meds with a very nice results and a very shallow bleb and a deep chamber. Currently, we are developing 2 new indications for MIMS. The first one is infranasal sclerostomy instead of going supranasal, where the 12 o'clock of the conjunctiva is already scarred from previous failed trabeculectomy cases. And the second indication is nasal trabeculostomy for mild glaucoma. Let's have a look on the nasal trabeculostomy procedure. We are creating 5 tiny holes into through the TM to the Schlemm's canal, as you can see here. The trephine here is going out only 0.5 millimeter instead of 2.5 millimeter. This is the second one. And now we will be able to see how exactly it's done via the animation. This is how it's done. As mentioned, we are doing 5 tiny holes. And this is it. This is the interim results of the trabeculostomy. You can see that the IP was dropped by 30% at 9 months and almost 0 medications at 9 months. These are the interim results of the infranasal sclerostomy. Remember, those are patients with severe glaucoma. After failed trab, the IP was reduced significantly, as well as the number of medications. The two new indications expand the potential market for MIMS to address almost all glaucoma patients who need surgery. We are supported by a distinguished list of experts from around the world, and we are heading to start, hopefully, the FDA study by the second part of this year. And I'm welcoming you to join the MIMS revolution. Thank you.
Dr. Grover, any comments?
Nir, very nice talk. Can you talk more about how, like, kind of the dimensions of that channel that's created, and how you know it kind of stays patent?
Sure. The dimensions are about 90 micron in diameter and 1 millimeter length. And why does the channel stays open? It's the first question that we were asked along the way. First reason that it stays open is because we are taking a piece of tissue out. We are not just getting in and out. We are just taking out a cylinder of tissue. Second, we use mitomycin to prevent the scarring from the outside between the sclera and the conch. And the continuous flow of the aqueous keeps the channel open.
I think Sean wants to use that little piece if you're not using it. No, I'm kidding. When you do the sclerostomy, you're just going through, you're not taking any tissue out for the nasal sclerostomy, trabeculostomy data that you were talking about. You're not removing tissue with that. You're just making a hole through the back wall of Schlemm's canal.
We are taking a piece of cylinder of tissue from the TM.
You are removing a tissue.
Yeah.
The same? Is
yes.
it the same? Okay.
To create a patent channel.
Well, we have a live question here we're very excited about.
Well, I don't know how much alive that is, but I'm Bob Stamper from San Francisco, and I have sort of two questions. One is that we've learned in the past that putting a hole through the sclera, full thickness hole, often ends up getting plugged with iris. And I was just wondering if any of your patients showed that. And the second question is, we've also learned in the past that putting filtering blebs down below risks potential for serious infection down the road. I wonder if you could just comment on those two things, please.
Yes, thank you for the good questions. So the answer for the first question is, yes, we had some iris plugging at the beginning, mainly for in phakic eyes, and to prevent it in phakic eyes, we recommend for iridotomy at the quadrant of the surgery, and we prescribe pilocarpine for the first few weeks after the surgery. And for your second question, we are not opening the conch, which is a huge advantage. It's done ab interno. We have a very shallow bleb, and this is one step before sending the patient for a tube.
Thank you. We're going to have to move on. And while our next speakers come up, Ray, I want you to speak on how is that different from the brown tree find. One thing about having Ray is he's seen most things. Our next speaker, by the way, is Sean O'Neill, who's the Chief Commercial Officer of ViaLase Incorporated. Ray, do you want to just...
Well, I think everything is just way higher tech than we did, to me, most of the same things, but you know, everything is much cooler with what Nir's doing. So I mean, there's plenty of things that didn't work, you know, in the Stone Age that work in the nuclear age.
Even just the elegance of the new sophistication.
You know, I mean, he's getting results that we never did.
Okay. Sean.
Thank you. I realize I have the dubious distinction of being the final talk here at this phenomenal day of innovation. I don't know if that means I'm batting cleanup or hurry up so that we can get to the final remarks and the reception. But very excited, and thank you to the Glaucoma Research Foundation for this opportunity. For me, one of the great pleasures is not just sharing the technology, but sharing the great work of this amazing team at ViaLase. The team actually is comprised of some world-leading engineers in the femtosecond laser space. Our CEO and co-founder, Tibor Juhas, was one of the original inventors of IntraLase, which revolutionized the refractive market, as well as LensX, which had a big impact on how, femtosecond laser cataract surgery. But really, the mission at ViaLase is solely focused on glaucoma, really redefining the treatment paradigm in glaucoma by unifying a micron-level OCT accuracy with the precision of the femtosecond laser. What's interesting with all technologies and innovation, though, is it's never an event. It's not a one-time occurrence. It's a journey. And this journey for the ViaLase team and Tibor and Ron Kurtz specifically was actually back in Michigan, where the first grant for femtosecond lasers was actually for a glaucoma application. Based on just certain aspects of the time, back in 1995, they actually refocused their efforts in the cornea and then refocused them again in cataract. But that desire to bring femtosecond lasers to glaucoma and the glaucomatous patient never waned. And in 2017, ViaLase was created, again, with that sole purpose, that laser focus, to bring together the precision of femtosecond laser and the micron accuracy of OCT. But obviously needed to prove it within the science, needed to prove that the technology was capable. And so in 2021, a human cadaver perfusion study was published demonstrating that, yes, femtosecond laser trabeculotomy treatment can significantly decrease IOP in this perfusion model. A second cadaver perfusion study was performed, again, demonstrating that this is the first direct evidence supporting the feasibility of clinically applicable, non-invasive femtosecond laser trabeculotomy for the treatment of glaucoma. What's also great about these publications is they demonstrated the hallmark of the femtosecond laser, which is its precision, which is its lack of negative effect on adjacent tissues or lack of collateral damage to adjacent tissues. So you see with the autofluorescence or transmission electron microscopy, where the TM is and then where the laser channel is created, there's no collateral damage to the adjacent tissues. That's led the team to create and design the ViaLux laser system, and really the ability to create and demonstrate the first femtosecond laser image-guided high-precision trabeculotomy or flight treatment. All the marketing people always love a good acronym, so flight rolls off the tongue a lot easier than those other words. But the design is, again, to take that micron resolution OCT, combine it with the femtosecond laser in the effort to decrease IOP by increasing aqueous outflow. We know in glaucoma surgery, though, visualization is so important. We've heard it on the panel many times. We've heard it throughout the day. The pristine gonio cameras, proprietary handheld gonio camera, as well as the ability to get these OCT images while the patient is non-invasively docked and the eye is coupled to the femtosecond laser, is a huge breakthrough in identifying not just trabecular meshwork, but Schlemm's canal, and ultimately the collector channels to really inform that surgical placement of that femtosecond laser trabeculotomy. So just to recap some of the key differentiators, micron accurate OCT, precisely identify the TM, Schlemm's canal. The femtosecond laser with its precision, the OCT imaging, non-invasively creating these channels. The channels, though, are programmable. Because the patient's eye is coupled through a patient interface, you have the ability to access all four quadrants of the eye versus being constrained by the location of your corneal incision. And then finally, there's no technical limitation to doing multiple channels. I'm going to share with you in a minute some data where we're looking at a single channel, but there's no technical limitation to doing multiple channels. So here is a quick video, and it's been edited for time. But there's the handheld gonio identifying the open angle where you want to treat. Now the patient is coupled to the laser. You see you can then utilize the imaging to identify and focus in the laser beam in the trabecular meshwork. Photo disruption now will occur to create that 500 by 200 micron channel. And that's what you see, the bubbles there, again, very normal in femtosecond laser. And then there's a really nice pristine channel. So again, share with you some of our pilot data. This is first in human data. So it was single case, single series. The real opportunity for this data was to assess safety. Obviously, we did observe IOP as well. No washout, single channel, unmasked. We did identify there were no adverse events at any of the time points out to 24 months. And we did identify that mean IOP was IOP reduction of about 32% from baseline at that 24-month time point, and 88% of the eyes achieved greater than 18 millimeters of mercury, or less than 18 millimeters of mercury. So just to summarize, again, here we have ViaLase focused on bringing the mission to redefine the glaucoma treatment through the introduction of the non-invasive ViaLux laser system, combining micron-accurate OCT with the precision of the femtosecond laser. Really encouraged by the 24-month data, which has informed our current RCT that we're enrolling for regulatory clearances for both CE mark and FDA clearance. And we look forward to continuing to update you on our milestones as we look to bring this novel technology to glaucoma patients worldwide. Thank you.
Thank you, Sean. We have a couple minutes for discussion. And Yvonne, you want to start the discussion?
That was really beautiful imaging to see that those OCT images and seeing the channels being created, as well as the histology to show that those channels are being created without any collateral damage. I'm wondering, though, still trying to understand after seeing the video, where does the where does the OCT fit in? Because it looked like you're just aiming the aiming beam for the laser, for the femto. But how does the OCT work? And then if you can describe to us a little bit also, because you mentioned treating different quadrants, is this being done in the clinic with the patient sitting up or reclined, or how is this going to work?
Yeah, so the design of the laser is to incorporate the OCT imaging to identify the trabecular meshwork to help guide the laser. So at this point, we are manually lining up the aiming beams, but ultimately the design is to leverage the onboard micron level OCT, but also in the ability to determine your treatment placement as well, so that you can ultimately identify where those collector channels are. Because one of the things that we hear quite often is the need in glaucoma conventional outflow pathway surgery is you not really know where the collector channels are. So if we can identify and place the intervention close to those collector channels, the sense is that you'll have a better opportunity for an IOP lowering event. So the OCT will be utilized in that manner as well. And then the location, you're correct, since it's non-invasive, non-incisional, it does not require an operating room environment. But so it would require like a laser room or a clean room. At this point, we believe that from a business model standpoint, it's most likely to be purchased by an ASC versus a clinic. Mm-hmm. So we anticipate it being in an ASC environment, but in a clean room or laser room environment.
Well, I think this wraps up our amazing 13-member panel over here, which is the largest I've seen in a long while. Thank you so much for joining us for the Emerging Glaucoma Devices. And thank you to our panel members and all the companies who have agreed to come up and share their technology.